Good afternoon. Welcome to MSK Science Spotlight. I am Danwei Huangfu. I am an associate member in the developmental biology program at Sloan Kettering Institute. This seminar series features lectures from today's leaders in basic and translational biomedical science. If you have questions throughout the talk, you can email them to the Office of Scientific Education and Training at oset@mskcc.org or by tweeting at MSK Education. So all those questions will be answered at the end of the program. Also a technical note, for the best viewing experience, please use Google Chrome. If you're using Internet Explorer, please use version 11 or higher. If you experience technical issues, please lower the video resolution. You can do so by clicking on the gear shift on the lower right of the screen. Now let me get to the important part. I'm pleased to welcome structural biologist Dr. Stephen Long, member of structural biology program at Sloan Kettering Institute. The Long Lab studies ion channels and membrane proteins using cryo-EM, X-ray crystallography, and functional approaches. His talk today is entitled, "And Now for Something Completely Different, Structure and Mechanism in the Calcium-Activated Chloride Channel BEST1." Steve?
Great, thank you very much for this opportunity to present some of our work today. It's really a pleasure to join you remotely. And I'm really interested in telling you about this story because this channel, Bestirfan 1, which is in our body, is really quite different from a lot of the other channels that we know about. And so I'd like to convince you of that through the talk using both structural and functional approaches. Before I jump into that, I'd just like to disclose that I have no competing financial interests, and also to acknowledge the three people that have really led this work. It was started by Veronica Kane-Dixon, who was a postdoc in the laboratory several years ago, and then carried on by George Vasey, who was in the Gerstner Sloan Kettering graduate program here at MSK and did his PhD thesis on this work, and then also joined by Alex Miller, who was a postdoc in the lab as well. So my laboratory is interested in trying to understand the mechanisms of eukaryotic ion channels that generate or respond to calcium signals. So these could be calcium channels themselves, like the calcium channel Ori, which is a calcium channel in the plasma membrane of cells. They could also be calcium channels in intracellular organelles, such as MCU, the mitochondrial calcium uniporter, which is present in mitochondria, and also calcium-activated channels. And so today I'll be telling you about Bestirfan, a protein located in the plasma membrane that's activated by the binding of calcium, and that opens an anion-selective pore through which chloride flows. Now, Bestirfan, we became interested in this Bestirfan channel because it defines a new family of chloride channels. And this was work stemming from Jeremy Nathan's laboratory that found that when he expressed this protein, Bestirfan 1, in eukaryotic cells, that you obtained a chloride selective channel. And so these are recordings, electrical recordings, electrophysiological recordings, where we're plotting current versus time. And you can see that cells containing Bestirfan have currents that can be elicited by voltage. And we became interested in this protein because it was calcium dependent. So you can see in the presence of EGTA, you don't have much current. EGTA is a chelator for calcium. But when calcium is present, you get a sizable amount of current. And the sequence of this protein was really very different from other ion channels. And so we thought it represented a new family of ion channels. And why is it called Bestirfan? Well, Dr. Best was one of the first people to characterize some of the mutation, or this an eye disease which is known as a BEST disease. It is macular degeneration, so you lose your central vision, and this here is a buildup of a material called lefuscin in the macula of the eye, and this causes a loss of central vision, which is usually a juvenile onset disease. It's fairly common, and there are over 200 mutations in this channel that give rise to this disease, and those mutations are depicted here as red spheres on the structure of the channel. And so you can see they're really pretty broadly distributed on the structure. But there probably are other physiological functions of Bestirfan proteins that really are not that well understood. It's a fairly enigmatic protein from a physiological perspective. We have 4 Bestirfan proteins in our body, 4 different types. They have fairly broad tissue distribution, which makes it somewhat harder to understand the particular functions of the channels. And as I mentioned, it's somewhat enigmatic. Some recent studies have identified that Bestirfan is required for volume regulation, and both in human retinal pigment epithelial cells and also in mice, in that case in the sperm of mice. But really a lot of the functions of Bestirfan are, I think, not well understood. And that was really interesting to us too, because in a way, the biochemistry, the functional studies, the structures that we did are in a way preceding some of our understanding of what this channel does physiologically. And I think that might be leading us into a better understanding of what it does physiologically. So before I begin to discuss Bestirfan and its particular properties, I'll just give a brief introduction to ion channels. And so this represents a membrane, a cellular membrane, and ions are essentially prevented from going through the membrane because an ion, such as depicted here as a red circle, has a charge, and the membrane is comprised of hydrophobic groups, and so the ions are essentially excluded from permeating through the membrane. And so we have ion channels or ion transporters that allow ions to flow, and this represents one of them. You can think of these as enzymes. They catalyze the flow of ions down their gradient, that is, down their both chemical or electrical gradient. So for example, if you have a solution that contains these red ions and these blue ions, and you have an ion channel in a membrane that is selective for red ions, those red ions will flow through. And so ion channels have 3 basic properties. They have the ability to allow ions to permeate. They catalyze the permeation of ions. They typically are selective, meaning this channel might be selective for a red ion, and they have the ability to open and close, which is depicted here by this gate, in this case, sort of a little flap. But this is really a conceptual idea. And sometimes gating is biased by the presence of a ligand, depicted here. So ion channels often have a region of their 3-dimensional structure, their 3-dimensional shape, that can be referred to as the selectivity filter, the part of the ion channel that's responsible for determining what type of ion goes through. And they have another region that's responsible for this opening and closing or gating process that could be biased by the binding of a ligand. So, and before I jump into Bestirfan, I thought I'd give a little bit of information about what is known about one particular type of ion channel, the most well-studied ion channels, I would say, or the most well-known ones, derived from ones that are related to potassium channels. And so this is work that goes back about 20 years or so, and from Rod McKinnon's laboratory. And potassium channels are essentially tetramers of 4 identical subunits. Sometimes they essentially have a 4-fold symmetry. And so here we're looking at the intracellular view of an ion channel from a bacteria, actually. This is just the ion pore. And if we look at now just 2 subunits, that's what's shown in the main panel here, spanning the membrane, these 2 subunits span the membrane. They have a region of their 3-dimensional structure that is responsible for selecting potassium ions. And so this is the selectivity filter, and we see ions binding within the selectivity filter. And then they have a region that's responsible for opening and closing, which we refer to as the gate. And this particular bacterial channel crystallized in an open conformation. And I'll just depict the kind of conformational changes that can occur in these channels. And so this is a closed conformation. So toggling back and forth between the open and the closed, you can get a sense of the kind of molecular rearrangements that occur in potassium channels. And they involve, you can see that the selectivity filter essentially remains fixed between these 2 conformations, whereas the gate goes from a very wide to a very narrow constriction. And this motion is accomplished by the bending and large motions of helices that line the ion conduction pore. And what I'd like to highlight today is that Bestirfan actually acts very differently, both in its terms of how it selects for ions and in terms of the kind of motions that are responsible for gating. But in summary, the narrow region often acts as a selectivity filter. You can see that this narrow region here often acts as a selectivity filter, and large bending motions are what open the gate. So we wanted to understand what are the mechanisms at work in Bestirfan for these three main properties that all ion channels essentially have. Bestirfan is anion selective, and it's dependent on calcium. So calcium binding somehow influences the gating properties. And so really most of the electrophysiological work that precedes ours, that really was the foundation of our work, was done by Chris Hartzell's laboratory at Emory University. And he found that these channels are monovalent anion selective, so they prefer -1 charges over -2 charges, for example. And they have this so-called lyotropic sequence of permeability, where, like many ion channels, different types of anions can go through. So in addition to chloride, ions such as thiocyanate, iodide, bromide can go through. And in fact, thiocyanate actually goes through more easily. And this so-called lyotropic sequence of permeability correlates with the ease of dehydration. So thiocyanate is more easily dehydrated than chloride, and it goes through more easily. And so that suggests in and of itself that as the ion goes through the channel, it must become at least partially dehydrated. And it was also known from Chris Hartzell's work that calcium somehow activates the channel, whether it's direct binding to the channel or through calcium binding protein. These weren't known, but it was clear that calcium has some effect on these currents that you observe. There was even a question as to whether or not this is a channel partly because of this lyotropic permeability sequence, or the ability of anion channels to permeate lots of different anions. It's often difficult to assign the permeation that you observe or the conduction that you observe to a particular ion channel that you're trying to study if you're not studying that protein in a purified setting, if instead you're studying it in a cell, in a cellular membrane. And so that was one of the reasons that motivated us trying to study it in a purified system. So really one of the things that we really set out to do was to determine the three-dimensional structure of Bestirfan. And so this is work done by Veronica Kane-Dixon that was published several years ago. And I'd like to give a shout out to the monoclonal antibody facility here at Memorial Sloan Kettering Cancer Center, and Frances Weiss Garcia in particular, because she helped us to develop these antibodies which bind to Bestirfan and allowed us to crystallize the protein. And so you can see that Bestirfan is a pentamer, 5 identical subunits. And here we're looking at the channel from the intracellular side. You can see that each of the antibodies binds to one of these subunits, and it has 5-fold symmetry. It was determined at about 2.9 angstrom resolution, which means that we can visualize all the amino acid side chains, and we can see calcium binding, for example. And this structure was determined in the presence of calcium, which made us think that probably it represents an open conformation of the channel. We presumed that it was an open conformation in the channel. And as the talk unfolds, you'll see that that was not quite right. But Bestirfan spans not only the membrane, so we're looking here at a side view of the channel. So this represents the portion that spans the membrane. Again, this is the cytosolic membrane. And then this large portion here would be located inside the cell. And so you can see that the channel really it sort of resembles a barrel. It almost looks like it has a portion of the channel that's embedded in the plasma membrane and a portion of the channel that's exposed and sort of dangling down into the cytosol. But they look like it looks like an integral thing, not a separate domain. Now there was this region that we could identify a calcium in, and we call this the calcium clasp. And so that started us thinking that perhaps this could be the calcium binding site, perhaps this channel is directly activated by calcium, and we'll come back to that. The channel is comprised of 5 identical subunits. Each of those subunits has a topology which is depicted here. You can see that each subunit passes through the membrane 4 times, and the regions of the polypeptide also extend into the cytosol to form this cytosolic region. And the cytosolic region is really an integral part of the channel. If you make a slice down the middle of the channel, what you reveal is that it contains a pore, which is the ion conduction pore. And this pore is about 95 angstroms in length. So it extends not only through the transmembrane region, but also far into the cytosol. And ions, if they were to pass through here, could not exit through side entryways or things. And so it's essentially sealed. This ion conduction pathway is sealed to the sides. And so an ion, as it moves through, would experience some different constrictions along the way. It would experience this aperture, a hydrophobic constriction near the cytosolic end of the protein. And then it would experience this larger inner cavity. And then the neck region, which is, again, a hydrophobic constriction, 3 amino acids from each of the 5 subunits, so 15 hydrophobic amino acids line this ion conduction pore in this section, and then the outer entryway. And so we named these the aperture and the neck of the pore, and then this calcium binding site is the calcium clasp. And really wanted to ask the question, now that we have the structure, what do these parts of the protein do? And where is the calcium sensor? Where is the gate, the part of the protein that opens and closes, like that triangle we were looking at earlier? Where is the part of the gate? Where's the part of the protein that opens and closes to prevent or permit ions from permeating through the channel? And how is ion selectivity achieved? Both a selectivity for anions over cations and selectivity among the anions. Remember, thiocyanate is more permeable than chloride. So before we jump into that, I want to introduce bilayer electrophysiology. And this is a technique that we use in the lab a lot to study ion channels. And it's I think, a very beautiful, very powerful technique because it allows us to study a purified ion channel embedded in a lipid membrane. And here I'm showing the experimental setup where there are 2 aqueous compartments that are separated by a partition. And in this partition, there's a small hole. This is not drawn to scale, but this small hole, onto the small hole, we paint a lipid bilayer and then insert a purified ion channel into that lipid bilayer. We can then, using electronics, control the voltage gradient across this membrane by injecting or subtracting current. And so we can also measure the current. Now you might ask, what is the current from? Well, the current is actually carried by the ions as they pass through the channel. So we can study individual channels or a collection of channels by measuring the ion current that's carried by the ions as they pass through this channel. So for example, chloride ions as they flow through the channel. And that allows us to really interrogate some properties of the channel. And this experimental system also allows us to control the solutions on either side of the membrane so we can really address some basic questions about how the channel functions. And so these are some electrical recordings made from a different ion channel just to highlight some of the things that you can do. So here we're plotting current versus time. And this is the so-called voltage protocol that we give it. So here the channel is closed and here the channel is open. This is looking at a single ion channel recorded in a membrane. You can measure the current through a single ion channel. You can also measure the current through multiple ion channels. And so here is the current that is elicited from, that's due to ions flowing through multiple ion channels. And what I'll be describing today is the technique of using multiple channels. And this is largely because it allows you to study, it's essentially a signal to noise issue. And so if you need to have a good strong signal, then it's easier to study multiple channels. But one of the things that we'd like to do in the future is to study single channels of Bestirfan. So what can you do with bilayer electrophysiology? So imagine that we have a gradient of 30 millimolar potassium chloride on one side of the membrane and 10 millimolar potassium chloride on the other side, and that we have an ion channel embedded in that membrane that is selective for, in this case, chloride. So you can see that if we could initiate this experiment, the chloride ions would flow in, they would flow down, and that would build up negative charge on the inside. We'll call this the inside, relative to the outside. And you can see that at some period of time, after enough ions have flowed, or if we control the voltage in some way, so this separation of charge creates a voltage. If we control this voltage to some extent, such that it's negative enough on this side, you can see that the net ion flow will cease because these negative charges repel the permeation of the ions through the channel. And the voltage at which this occurs is called the reversal potential, because if you increase this voltage, make it more negative, the ions will actually flow backwards in a way, and so they will reverse. And so the reversal potential, the voltage at which there is no net flow, is a measure of ion selectivity, and that's something that we can measure. So here, for example, we're plotting current versus voltage, and this reversal potential is about 25 millivolts for this Bestirfan channel, which indicates that it has what's called a permeability ratio. So a permeability ratio is a way to quantify ion selectivity. It represents the permeability of one ion relative to another. So in this case, potassium versus chloride, chloride is about 20 times more permeable than potassium. And this is obtained through the Goldman-Hodgkin-Katz equation. So this again is sort of to highlight the experimental setup. And as the talk goes on, I'll try and indicate the experimental setup with a diagram like this. And so in this case, we have an ion channel in the partition, and the two chambers contain a different concentration of potassium chloride, and we can arrive at our reversal potential. These are the actual recordings that we make. And so we conduct an experiment where we step the voltage, for example, from 0 millivolts to 100 millivolts, and then we record the current. And so that's what this top trace represents. So this is a collection of data that's all a collection of experiments that is represented on one figure, and then this is a collection of data that results from that. But here we're looking at current versus time. And so you can see that if you stepped 100 millivolts, you get some amount of current. And then we can take a window of this section here and then come up with a current versus voltage relationship, and then arrive at the reversal potential, which again is a measure of ion selectivity. And that shows us that this channel, this is purified Bestirfan now in this system, and that shows us that this channel is selective for chloride. If this channel were not selective for chloride, but allowed potassium to permeate just equally as well, the reversal potential would be 0 millivolts. And so this shift from 0 to 25 millivolts is a measure of ion selectivity. Okay, so Bestirfan is chloride selective, and similar to what Chris Hartzell had shown using whole cells. Purified Bestirfan is also calcium dependent. So if we have calcium present, we see currents. If we add EGTA, which chelates the calcium, those currents go away. So Bestirfan is calcium dependent, which means that it's directly activated by calcium binding. It's not binding through some other protein. And we can titrate back calcium and arrive at about 17 nanomolar K1 half. And this again is purified protein. And purified protein also has this lyotropic permeability sequence. And so this is an experiment where you have potassium chloride on one side of the membrane, and then say potassium thiocyanate on the other side of the membrane, potassium iodide, and can do different experiments. And what you observe is that thiocyanate has a reversal potential about minus 60 millivolts, which means that it's more permeable. So the permeability ratio is about 10. So thiocyanate is about 10 times more permeable than chloride through this channel, which is kind of interesting because thiocyanate is actually bigger than chloride. It's a bigger molecule, but it's more permeable. But again, this is probably because of the dehydration energy. Thiocyanate is more easily dehydrated than chloride. Okay. So far, we have basically just recapitulated what has been known about the channel to some extent from whole cell patch clamps, but it shows for sure that Bestirfan is an ion channel and it's directly gated by calcium. And I should mention that we've been studying chicken Bestirfan. Now you might ask, why are we studying chicken Bestirfan? Well, it turned out that we cloned about 30 or so different Bestirfan proteins, and chicken was the most stable among those that we cloned biochemically. And this really this is an important tool because we can study the function of this more easily if it's more biochemically stable. But these experiments that I've just shown you show that chicken Bestirfan behaves very similarly to human Bestirfan-1. It's about 74% identical in its amino acid sequence, and it has anion selectivity, the lyotropic permeability sequence. And so the conclusions that we're reaching about chicken Bestirfan-1 really apply to the family of Bestirfan channels. But now that we have this methodology developed, what can we do to try and understand the structure and understand the regions of the protein that we refer to as the neck, the calcium clasp, and the aperture, and what they do? So first, I'd like to address the function of the neck. And so the neck, again, is comprised of 3 amino acids from each of the 5 subunits that are hydrophobic. So we did a very simple experiment, just replace each one of those amino acids with an alanine. So in that case, it's 15 amino acid substitutions within the context of the pentameric structure. Now, this is the wild type data that I have already shown you, reversal potential of 25 millivolts. When you make this NEC mutation, you also get a reversal potential of 25 millivolts, which means that the NEC is not responsible for selectivity of chloride versus potassium. And this is really, was actually pretty surprising, because if you look at the structure, the NEC is a very narrow region. It actually looks somewhat similar to the selectivity filter in potassium channels in terms of its dimensions, not so much in its chemistry, but certainly in its dimensions. But we find that the NEC is really not a determinant of anion versus cation selectivity. We get the same selectivity for anions in this context of this NEC mutant. Well, you might say, well, maybe it's affected the structure, but we've actually determined the structure of this triple alanine mutation, and in fact, it's just replacing, it's essentially just replacing each of these side chains with an alanine, which would widen the NEC. What about selectivity among the anions? Do we have any, is there any effect on this lyotropic permeability sequence? Well, with the triple alanine mutation has the same lyotropic permeability sequence, and so it does not affect permeability among the anions, which is also pretty surprising, because this is a narrow region. You'd think that ions would have to be dehydrated as they move through, and so you might imagine that making this wider might have some effect here, but it didn't have really, it had no effect. You can see that these are basically identical. So it did not affect anion selectivity. Okay, what about the other main function of the channel, which is calcium dependence? Well, it turns out that this has a dramatic effect on calcium dependence, this NEC does. So again, the wild type protein is calcium dependent. Here's in the presence of calcium currents. In the absence of calcium, they completely go away. You can represent that on an IV curve like this. But you can see in the context of this triple alanine mutation that EGTA essentially has very little effect on the currents. And so that starts to suggest that this NEC might be involved in calcium gating, and that the NEC might be the calcium dependent gate. That is the portion of the channel that opens and closes through some molecular motion that either allows or prevents ions from flowing through the channel. And through work that is published, and I'm not going to go into at the moment, we identified that this calcium binding site that we observe on the structure is actually the calcium sensor. So the calcium clasp is the calcium sensor. If we mutate the calcium sensor, the channel is constitutively closed. As I mentioned, if we make this alanine mutations to the NEC, the channel is constitutively open. And if we combine these mutations, the one that knocks out the calcium binding site with the one that makes the channel constitutively open, what we find is that the channel is constitutively open. So if you knock out the calcium binding site and make the mutation in the NEC, the channel is constitutively open, which strongly indicates a pretty good indication that the NEC is the gate of the channel, the part of the channel that opens and closes to control anion permeability. So in conclusion, the NEC has no, for this portion of the talk, the NEC has no apparent role in ion selectivity. This was really a surprise, but in fact, the NEC is a calcium-dependent gate. So this indicates that Bestirfan is very different than potassium channels. In potassium channels, this is the selectivity filter, and this is the gate. Well, it turns out this is, the NEC is the gate in Bestirfan, and we don't yet know where the selectivity filter is. But I'd like to draw our attention to the aperture. What does the aperture do? And so we did a series of similar experiments. Just the aperture is lined by a single valine residue from each of the 5 subunits. And so mutating this valine to alanine, a simple experiment, could have a potentially dramatic effect. And so this is a summary of some of that data. So you can see that in the presence of calcium, you get current. In the absence of calcium, the current goes away. So unlike the NEC, mutating the aperture has no effect on calcium dependence. So that's a pretty good indication that the aperture is not a calcium dependent gate. And you can see also that it reverses at 25 millivolts when we have a gradient of 30 versus 10 potassium chloride, which indicates that the aperture, is not a determinant of anion versus cation selectivity. So the aperture is not responsible for anion versus cation selectivity, and the aperture is not a calcium-dependent gate. What about our lyotropic permeability sequence? Well, it was incredibly dramatic. The lyotropic permeability sequence that we observed for wild type is completely gone in the context of this aperture mutation. So this is really amazing, because what this means is that the channel is actually very different than a potassium channel, because remember, this channel is still selective for chloride over potassium. but it's not selective for chloride versus thiocyanate. There's no difference there. So that means that different parts of the channel are responsible for anion versus cation selectivity, then are responsible for this lyotropic or selectivity among the anions. So taking this a step further, we did this experiment where we took the same setup and we replaced one of the sides with acetate, so potassium acetate versus potassium chloride. And what you find is that acetate can actually flow through the mutant channel. It doesn't flow through the wild type channel, but it can actually flow through the mutant channel, which means that the aperture, when we make this mutation to the aperture, that you can get really large things such as acetate to flow through the channel. Okay. So that suggested to us that the aperture is a size selective filter. It's governing what kind of thing goes through there on the basis of size, and it lets small things go through. So in conclusions about the aperture is a size selective filter. It gives the channel its lyotropic permeability sequence, but it's really not a calcium dependent gate. Okay, so this is where we are now. So we understand that the neck is the calcium dependent gate. This is the calcium sensor. This gives us some measure of selectivity. But one of the things that was really, that we were really wondering about is, okay, we have this structure. What does this structure represent? We have a very narrow neck region. We've shown that the neck is the calcium-dependent gate, but the neck looks very small. It's a very thin, it's a very narrow constriction, and it would be hard to imagine how an anion could get through there. Perhaps it could get through there dehydrated, but that wouldn't make too much sense because we know that the neck is not responsible for the lyotropic permeability sequence, which is an indication, of the dehydration process that's going on. And so it really made us start to wonder, well, maybe we don't have an open conformation of the channel. Maybe this represents some other conformation of the channel. And then some work that was done by Chris Hartzell's laboratory again, we came back to this, and where he had showed that if you look at currents through Bestirfan over time, they decrease. And he called this current rundown because the molecular process wasn't well understood. But the currents would go away. You'd see some current and it would go away. And the rate at which it went away or decreased was dependent upon the calcium concentration. So this was calcium-dependent current rundown. But the mechanism was really unclear. He did find that it involves phosphorylation of a C-terminal peptide. So there's this portion of the channel, a C-terminal peptide, that seemed to be important in this process. And so we set out to try and understand if we could recapitulate this in a purified setting. Again, the experiments that were done by Chris Hartzell were done in a whole cell context and could, for example, be due to the diffusion of a ligand away from the channel that's necessary for its function. But in a purified setting, what George Vasey observed is that you can get current decrease that's dependent on the calcium concentration. So here we're looking at currents through the channel over the course of time, and you can see that there's a decrease throughout time that's dependent upon the calcium concentration. Now this decrease is reversible. So now we know that calcium-dependent current rundown is due to a process that we call inactivation. The channel opens, it allows ions to flow through, and then after some period of time, the channel closes. Now channel inactivation is a very common process among ion channels, but as I'll show you, the mechanism for Bestirfan inactivation is really quite different than other ion channels. So I haven't mentioned, but you can imagine there are 5 subunits here. The blue subunit has its C-terminal tail, wrapped around 2 adjacent subunits. And this portion of the polypeptide is the portion that Chris Hartzell's laboratory identified that might be involved in what we now know as inactivation. And so George did a series of experiments, but one of which was just to cut off this peptide by truncating the protein on amino acid 345. And you can see that now the channel no longer inactivates. We no longer see a decrease in the current through the channel, even at very high concentrations of calcium. It turns out that you can make mutations to this peptide. You can also make mutations to the site in which it binds. And that led us to the idea that this is an inactivation peptide that binds to a cell surface receptor, it binds to a receptor on the cytosolic surface of the channel. And mutations can disrupt its binding and its effect on the function of the channel. But where might be the inactivation gate? How is this channel inactivated? Why are currents prevented from going through the channel after some period of time? I guess before I jump into that, I'll just mention that the Bestirfan 345 construct, the one that no longer inactivates, is otherwise very, very similar to the wild type one. It's dependent upon calcium. It's selective for chloride over potassium, and it has a very similar activation for potassium. Okay, but it turns out that if you mutate the neck, we go back to our alanine mutation of the neck, this channel also no longer inactivates, even in the context of a full-length protein here, in the context of a protein that contains the inactivation peptide. So that suggests that the neck is also the inactivation gate. And so the neck is the inactivation gate, and it's governed by the binding of this peptide to the surface of the channel through a receptor. Okay, which is depicted here. So the binding of this peptide to a surface receptor on the surface of the channel controls inactivation. And how does it do so? It must do so through an allosteric means, because this peptide is nowhere near the neck. It's on the surface. It's on the outside part of the channel. It's not physically occluding the pore, but somehow there's an allosteric mechanism wherein the binding of this peptide causes this neck to close. And so we now started to think that the structure that we had might represent a closed conformation or an inactivated conformation of the channel, even though calcium is bound. And this was further supported by the observation that the antibody that we use to determine the structure seems to stabilize the inactivated conformation. So if you record currents under conditions where they wouldn't inactivate very much because you have a low concentration of calcium, and then you add the antibody, these currents rapidly go away. So that suggests that perhaps the antibody is stabilizing the inactivated conformation, and so that the structure probably represents an inactivated conformation of the channel. And what would the open structure look like? Well, this is where cryo-electron microscopy came into the picture, and we were able to determine a cryo-electron microscopy structure of Bestirfan in the calcium-bound inactivated state, the same conformation that we had previously, and then try and figure out ways to prevent inactivation. And we knew we could remove that peptide. We had other antibodies actually that prevented inactivation. We could mutate the binding site for the peptide. You could mutate the peptide itself. We tried a lot of these different approaches, and the approach that worked was just to remove the inactivation peptide. And so we just chopped it off here, the same construct that I had mentioned previously that George investigated. And what that allowed us to do is to observe two different conformations of the channel, which were really quite amazing. These were both present in the cryo-electron microscopy sample. So this is the same protein, purified, imaged on the microscope. And what we observe is that we see two different conformations of the channel. One is a closed conformation. This is looking now at a slice through the neck region of the channel. One is a closed conformation, and one is a dramatically open conformation of the neck. So this started to make sense. The neck undergoes a dramatic opening under certain circumstances. And both of these structures are determined about 3 angstrom resolution. And looking at the size of the ion conduction pore, you can see the dramatic change that occurs as the channel moves from a conformation through which ions cannot flow to a conformation through which ions can flow, the open conformation of the channel. Now in the sample, this represented only about 10% of the particles, only 10% of the channels were in this open conformation relative to this closed conformation, but you can see this dramatic motion. And I'd like to highlight this dramatic motion because it's really quite different than the types of motions that we were looking at for potassium channels. And so this is a depiction of the closed conformation of the neck. Here we're looking at a slice through the neck where these red amino acids are the hydrophobic amino acids that line the neck. And then these green ones are hydrophobic amino acids more in the middle of the protein. And so this is going to be a morph, just a continuous extrapolation between the two structures that we have, the closed conformation and the open conformation, but it'll highlight the conformational changes that are occurring. So that's open. and closed. This is really one of the most amazing conformational changes that I know about in an ion channel in terms of the molecular rearrangements that occur when the channel goes from a non-conductive to a conductive conformation. And you can see that it actually doesn't involve it doesn't really involve the same kind of motions that occur in a potassium channel. It doesn't involve helices that bend out of the way, but instead it involves a series of side chain rearrangements, a concertina of side chain rearrangements that are propagated throughout this middle section of the channel. So it's these side chain rearrangements that govern the opening process. And there is an outward motion of those helices a little bit as well, and a slight rotation of the helices. But and a feature of the channel that was really quite striking to us when we first looked at the structure, if you look at the helices that line the ion conduction pathway, this green one here, you can see that it has a continuous helix part, and then it has these broken sections on the N and the C terminus of that helix. Now this represents the transmembrane region about right here. And so this helix is broken right at the middle of the membrane, which is really kind of an unusual feature for an ion channel. And it turns out that this helix essentially is sort of tethered. It's sort of floating. Tethered helices permit the opening process, permit this concertina of amino acid rearrangements that allow the channel to go from this narrow closed conformation to this wider, really widened open conformation. And these molecular rearrangements involve side chain rotamer changes. They actually go from the most prevalent side chain rotamer to the second most prevalent for all of these amino acids. And in doing so, they actually expose these blue amino acids, which are serine ones. And so the pore then becomes hydrophilic. So what is the coupling? How does calcium binding somehow instigate this process? And one of the things that we noticed is that there's a tryptophan residue here that undergoes this molecular rearrangement. And so we just asked a simple question, well, what happens when we mutate that tryptophan? And so Alex Miller mutated that tryptophan to a phenylalanine and determined the structure. But what she found is that this simple mutation, which does not line the pore and is not in the calcium binding site, prevents the channel from responding to calcium. So in the absence of calcium, you get current. In the presence of calcium, you get current. So it's behaving sort of like the mutations of the neck, but this is essentially locking the channel in an open conformation. And so when she looked at the structure, in fact, that's exactly what you see. The channel is locked in an open conformation. So this residue is involved in in allosterically controlling through space how the channel opens. So you might ask, well, what happens in the absence of calcium? And so Alex Miller did the experiment where she carefully removed calcium and determined the structure. And what you find is that calcium clasp, the part of the protein that binds calcium, is essentially disordered, but that otherwise the structure looks identical to the closed conformation. And so what that means is that the same conformation of the neck, we really observe only two conformations of the neck, either the closed conformation or the open conformation. And putting all this together, we have three different structures: the calcium-free closed, the calcium-bound closed, and the calcium-bound inactivated, that all have the same conformation of the neck, which indicates that both activation and inactivation gating have the same kind of motion. It's the same molecular rearrangements that are responsible for these two different processes, and that we really only have one open conformation, which is shown here. And so now we have structures that represent all of the major steps along the gating cycle and can propose a model for gating. So starting from the calcium-free closed conformation of the channel, calcium is not bound, the neck is closed. Calcium can bind, but the neck can still remain closed. And then over some period of time, or it can toggle back and forth between this open conformation in which the neck is much wider, or the closed conformation in which it's narrow. And so this is an important thing to remember, because ion channels, the binding a ligand doesn't necessarily lock it into one conformation or another. It biases the equilibrium between these two different conformations. And then once it's open, the channel can then inactivate by the binding of this peptide to the receptor on the surface of the channel, and then arrive at the calcium-bound inactivated state. So now I'm wrapping up the talk, and as we think about the open conformation of the channel, this really was a shock to us, just how wide this section of the pore is. And the only constriction then is the aperture. Really large things can fit through here if it weren't for the aperture. But we know that the aperture is responsible for the kind of things that go through there, because if you mutate that aperture in the chicken channel to an alanine, then you no longer have discrimination among the anions, and even things like acetate can go through. And so, and here's George made several other mutations. So alanine, glycine, you can see the acetate, propionate, butyrate. These large anions can actually flow through the channel if you make these mutations to the aperture. But if you look at the sequence of Bestirfan channels, they actually vary a lot at the aperture. In fact, the aperture is one of the regions that they vary the most. And so chicken Bestirfan 1 has a valine, human Bestirfan 1 has an isoleucine, but human Bestirfan 2 has a serine at this location. And so these changes we think are going to have dramatic effects on the kind of ions that can go through. And that really starts to raise the question of, well, what kind of ions can actually go through? Could it be other things like the neurotransmitter GABA, which has been implicated in flowing through this channel? And I should also point out that Ting-Ting Yang's group recently determined the structure of Bestirfan 2 from the bovine, and she finds that the aperture is actually different in its dimensions there. So in summary, we have dissected some of the basic properties, some of the basic functional properties of this channel, identifying the neck as the constriction that governs calcium-dependent gating. So this is both the activation gate and the inactivation gate that's biased by the binding of a calcium to the calcium sensor through an allosteric mechanism, and also by the binding of an inactivation peptide to the surface of the channel. And the aperture, the neck is not responsible for ion selectivity. There's no aspect of ion selectivity that we can detect that's affected by the neck. On the other hand, ion selectivity is affected by the aperture, and mutating the aperture allows different kinds of ions to flow through. But interestingly enough, the channel is still selective for chloride over potassium. And so we think that chloride selectivity comes from different parts of the channel, which I haven't mentioned, but are indicated by these binding sites for chloride ions. So with that, I'd like to really thank the three people who have worked on the project, Veronica, George, and Alex, who have all moved on. And I'd really like to acknowledge my current laboratory and David Gatsby at Rockefeller University. The late David Gatsby was really a wonderful colleague and someone who we could go to with electrophysiology questions, and was a mentor to George. And so we really acknowledge his help and just contribution to this work as well. And we thank the NIH for their support of the project. And I'd be happy to take any questions.
Thank you, Steve, for a very informative talk. I think I can't believe it. Even I can follow the structural biology talk. So I'll start with maybe a question of my own naivety. So about the aperture, actually the inactivation peptide. I wonder, how does it actually get to the position to inactivate the channel?
Okay, yeah, so the inactivation peptide it's very interesting. It's part of the channel. It's the C-terminal part of the polypeptide. So it's essentially dangling around. And we've shown that, for example, if you remove calcium, that the peptide is much more susceptible to proteolysis. And so the peptide can be easily cleaved off if there's no calcium bound. And so we actually think that, as I mentioned, there's a calcium dependence to inactivation. And you might ask, well, how is that? We think it's actually not through the calcium sensor binding site, but through another calcium binding site on the surface of the channel, actually, that affects the binding of this inactivation peptide. And so I think, in a way, the inactivation peptide is really one of the more functionally important regions of the protein, because this varies a lot also between the Bestirfan proteins. It's also phosphorylated. There's a serine residue that Chris Hartzell's lab has shown to be phosphorylated. And if you if you mimic that phosphate by adding a glutamate in position in place of the serine, then you can prevent inactivation. So I think the is essentially tethered and ready to bind to its receptor, which probably means it doesn't have a very high affinity for its receptor. And in fact, when we have tried to do experiments where we add it exogenously, we can't see any effect. And so I think it's tethered. Being tethered allows it to bind.
I see. So actually, from the audience, there are some related questions. I think one of the questions is about this inhibitory peptide, whether it also has an effect on calcium affinity or cooperativity for the calcium binding.
Yeah, no, that's a great question. So in fact, it doesn't, as far as we can tell, and I'll see if I can go to that figure. Yeah. So here we're looking at the wild type channel or the channel missing that peptide. And you can see that they're both equally selective for chloride. They have a reversal potential of about 25 millivolts. They both are dependent upon calcium. So if you remove calcium, the currents go away. And you can titrate back calcium and then come up with a K1/2 of about 4 nanomolar for the one missing the peptide, about 17 nanomolar. But even those numbers are, it's difficult. You know, you can see we don't have very many titration points down here. It's hard to control calcium such a low concentration. But as far as we can tell, it doesn't affect cooperativity for calcium binding, but there could be a cooperativity among the binding of the peptide itself. We do certainly think that there is cooperativity in calcium activation. You know, one of the things that I didn't, I mentioned several times how this is a calcium dependent channel, but I didn't really tell you about why is it calcium dependent or how it's calcium dependent. What I showed you is that when calcium is bound and when the inactivation peptide is not inactivating the channel, that the channel can occupy an open or a closed conformation, and that when calcium is removed, the channel is always in the closed conformation. So we think the binding of calcium shifts the equilibrium such that you can occupy an open conformation some fraction of the time and a closed conformation some fraction of the time. And that's something that we'd like to investigate further using single channel measurements, where we measure the current that's carried by ions as they flow through a single Bestirfan channel. And certainly I think there's going to be cooperativity in calcium binding to the calcium sensor.
I see. So that's maybe also related to another question from the audience. So basically, this audience is asking, what prevents this inactivation peptide from inactivating in the absence of calcium?
Okay, so maybe I'll go to a slightly different slide here. And so I think so a lot of channels undergo what can be called as calcium-dependent inactivation, where higher concentrations of calcium cause the channel to inactivate through different mechanisms, actually. And this, I didn't point it out, but this mechanism is very different than other types of mechanisms. But remember that calcium activation, the concentration of calcium that causes the channel to open or biases the open conformation, is in the 10 nanomolar range. Whereas the concentration of calcium that causes inactivation is in the 50 micromolar range. And so these are different processes. And this disparity in these binding affinities or concentrations in which you get these effects really indicates that these are different processes. If they were closer in binding affinities, then they might be more related, but in fact, they're actually different processes. And we're starting to think, we might have identified, you can make mutations on the surface of the protein near where the inactivation peptide binds to amino acids that are negatively charged, and you can actually affect some of this calcium dependent inactivation. So we think that they're actually different processes.
Mm-hmm. So the next question is more related to physiology. So I think in the beginning of your talk, you mentioned BEST1 is mutated in human disease, macular degeneration, and lots of mutations were found. So can you comment on where were those mutations found?
Yeah, absolutely. So I'll just toggle back to that slide. Okay. Well, you can see that they're all over the place. And that's kind of interesting. They're certainly highly present in the principal components of the gating apparatus. That is the calcium binding site here. You can see that every single amino acid surrounding the calcium binding site, also in this region of the protein, is associated with disease. There are mutations within the neck. That's also a very highly prevalent region. That tryptophan that I mentioned is involved in coupling is also a site of mutation. And some of these have been studied in terms of what effects they have on the function of the channel. A lot of them you don't get currents. Some of them you do get currents, but they're altered in the sense that maybe they don't respond to calcium anymore. Certainly, if you mutate any of the calcium binding ligands, you're not going to have a channel that ever opens. But you know why this causes eye disease is really not clear. These are juvenile onset, so you know people are born with this mutation, but they don't develop eye disease until adolescence. And so it's not directly involved in neurotransmission, for example. It has more to do with the buildup of this lefuscin material. It's essentially occluding, you know, a damaging part of the macula. So, you know, I think that it's a hint as what it might be doing, but it's not directly involved. It's interesting, if you make the knockout mouse of Bestirfan 1, mice don't have a macula, and they have no apparent visual impairment. They do have much lower fertility, which turns out to be due to the inability of the sperm to withstand, or the reduced ability of the sperm to withstand osmotic shock. And so the sperm actually begin to swell, and that's responsible for their lower fertility. And that's that might be pointing to a very important physiological function of this channel, to prevent cells from rupturing when exposed to low salt environments, because water flows through membranes very easily, but ions don't flow through membranes. And so you have to have ion channels that allow the release of ions when cells like epithelial cells are exposed to low salt environments, which of course happens a lot in our body.
Our last question is also related to physiology. So the aperture, you mentioned that it controls anion permeability. So basically it's selective among different anions. But most of us think about the channel as a chloride channel. So what are the physiological implications for the cell, in terms of that kind of selectivity?
Yeah, no, that's a great question. So it certainly is a chloride channel in the sense that chloride goes through it. But like a lot of anion channels, other things probably go through it as well. So nitrate probably goes through it. Carbonate certainly goes through it. And we even suspect that larger anions, on the order of the size of acetate might go through it, or even other types of molecules. Because remember, the channel is actually 20-fold selective for chloride over potassium. So potassium actually goes through this channel pretty well. And it may do so in physiologically important ways. And so, for example, a neurotransmitter like GABA, which has been implicated to go through this channel by Justin Lee's laboratory in a series of studies, could actually permeate through the channel very slowly, maybe not quick enough to record in any kind of electrophysiological setting. Of course, GABA is also a zwitterion, so that makes that difficult. But it may go through very slowly. If it goes through 1,000 times more slowly than an ion, it may have an important physiological function. And so that's where that's one of the directions that we'd like to go. What are the physiological functions of Bestirfan channels? And I think that's one of the neat things that the structure is leading us toward, in the sense that, here we've started to understand how this channel works on a molecular level, but what is it doing physiologically? And that's definitely one of the areas that we'd like to explore.
Thank you so much for a great talk today. I would like to announce our next seminar happening on Monday, June 22nd. We are pleased to welcome Alex Joyner from my own program, Developmental Biology. The title of her lecture will be Constructing the Brain, Ensuring Plasticity and Repair. Thank you everyone for joining us.
Thank you.